Thank you very much for the very kind introduction. It's really nice to be here, and I very fondly remember when I was in high school how much at that time people influenced my career. My biggest influence in my career was my father, who, since I was 3-4 years old, taught me mathematics. And while I didn't understand really what he's doing in engineering, but we had a common language, it was mathematics. And today I'd like to talk to you also about the common language, but this is for experimental scientists. I'm an experimental scientist, not a mathematician. It's my hobby, but it turns out that I'm doing experimental science since 1995, so quite a time. And turns out there are common elements that I see in my own lab or students, that how we proceed or actually reach results, and how things can go wrong. So I'd like to share my experience with you, and I call them the elements of scientific discovery. And I will talk about 6 elements of scientific discovery, and I will illustrate you with one of the projects you heard about. Is like restoring vision. It's a long-term project, and one can never fully succeed because you can always do better. But nevertheless, this is something that is running in my mind and in my hands for 20 years. So this is the menu for today. I like to talk about the 6 basic ingredients of scientific discovery, illustrated by one project that took 20 years and it's ongoing. So I'd like to start with number 1. With where everything starts, and that is thinking, asking an important yet answerable questions. And there are 3 important here. One is thinking, the second is important question, and the third one is answerable question. So I like to talk a little bit about my science, and then I'll introduce you why I think thinking was required at that time, why I think the question was important at that time, and why I think even at that time, 20 years ago, I had a feeling the question is answerable. In practice, what happens is that many of you start your PhD somewhere. And initially, you can ask all sorts of questions, but they are not grounded in reality. But you spend some time in a graduate school where you kind of start to know your field, your laboratory. At one point in time, you have some understanding when you actually can raise an important question. So this is exactly what happened to me. By complete chance, I met someone who studied vision. It was complete chance. I didn't want to study vision. There was a dinner, and my father and another scientist who has studied the retina and kind of talked to me that the retina is the most amazing thing in the world. This is obviously not true, but I was young, and I get very excited. So vision starts with a video. That video enters our eyes. It is captured by the retina, and then it sort of moves forward. There is an important station, a very, very important station. Very little we know about what is happening there. It's called the LGN. And then it arrives the information to the back of your brain, into a visual cortex, and it radiates through many parts of the cortex. In a human, around half of the cortex, you can record visual information. So we are very, very visual animals. And there is something very unique and interesting about, so my laboratory basically studies these 3 stations. And something very interesting about the retina in this whole pathway, it's a really tiny part of the visual system. So when you see, you much see with what the cortex is telling you, but almost all visual diseases originate in the eye and not in other parts. So it's a tiny part, but for medicine, basically that's it. If you understand the retina and design therapies, you can cure 95% of the diseases. So that was kind of the original idea. And the other one is the retina is really a machine. It's an understandable machine because the brain doesn't talk to the retina talks to the brain. So if you isolate the retina and peel it off, it's doing the same thing that it's doing in a normal eye. So you can actually study. It's an input-output system and perfectly understandable. So that was a kind of a motivation to study it. And as I told you, I started graduate school. I went in a lab where I had first no clue what is happening. But around 5 years later, I made some discoveries. Namely, I bumped into a new structure. I discovered that the retina is really working differently than other people imagined. It is a biological computer that one video comes in, but at least 20 or 30 videos come out from the retina and on the optic nerve going towards the brain. And this was, at that time, a very surprising discovery. Very few people believed in it. It took a torture to get published it in Nature 2001. But this was just a kind of a view on the retina, what it does, but more on that, we started to look into the retina and get a mechanistic understanding. So how is it computer doing it? And what you see that the retina is indeed not as a digital computer you have in your cell phone or wherever. In that digital computer, every element is the same. And the retina is a biological computer that elements are fundamentally different. We call them cell types. So here you see some drawing from Rahamoni Cajal, and each of these cells is a type because they are repeated many times. And they are kind of wired together to do computations. And at that time, when I finished my PhD, I recorded almost every day. So I had a schedule that I started around 5 o'clock in the morning and recorded, let's say, until 2. And then in the afternoon, I analyzed my data. And so doing it for 5 years, and at one point, I really understood every single cell what they are doing in the retina simply because I have seen them. So in the retina, you show light stimuli, you put an electrode in the retina, and you see what the cells are doing. Okay, so it was a really fun game. And the reason it was fun game, because at the end, somehow I concluded that, all sorts of scientists, they have their opinion. It's important, not important. But I realized that it's not very much worth of listening to others, what they say. But I know the retina. I can listen to what the retina is telling me, and that is actually what the biology does. It was a very early kind of satisfaction that, famous retina scientists told me this, and I knew it's just not true. I mean, I'm recording from this retina. So one of my deep first experience was that listen to the data and what biology tells us and not what other people told us. But nevertheless, at that time, there was a knowledge that developed that how the retina is working. We get a hands-on experience. And the really question was that what is the next step? So where you go from there? You can always study a little detail of what this cell type would do, but this is not very satisfactory. And I got quite lucky because this was exactly the time, around 20 years ago, when 3 German scientists, Georg Nagel, Peter Hegemann, and Ernst Bomberg, who were studying algae. And today's politicians, none of them would give you money to study algae. But why on earth? What is the application for that? But science doesn't work that way. Science just works that we are curious about the world, and we try to understand anything. Indeed, I very firmly believe that science for us, for an individual, is very similar to eating. We just need that. And some people call science, some people call just, we want to understand things. Who doesn't want to understand things, right? It's every human wants to understand things. And some of us, we call it science, but you can call it anything. It's this deep drive to understand. And these scientists were very interested how on earth algae can sense light and use this to navigate. And they discovered that algae are fundamentally different from, let's say, my photoreceptors. In a sense, they need a single gene to give a light response to the cell. The algae has one cell, so it's an easy one. And they were able to show now they take this gene and put it into another cell, and that cell again becomes light sensitive. And the key discovery was that a single cell is able to, a single gene product, a protein, is able to sense light. It's a channel, it's a simple device, opens and current flows. So very simply, you can transfer the property of light sensitivity to one to another one. And that was the day when I knew about the paper a bit before, finishing my PhD, I realized that now there is a question actually that I know how the retina works, really, really in detail. And there are people telling me that, okay, here, I have a single gene that can make things light sensitive, and I want to put the two together. And the reason is very simple, because there are blinding diseases where the photoreceptors are dysfunctional. So there is a movie going to your eye, but nothing going to the brain, but the computer is intact. There is nothing wrong with the retina computer. All what it is, it loses light sensitivity. So the idea is completely trivial at that time if you know the retina and you know there's a gene, And then start a more difficult part, of course, from idea to get to reality. And here is the idea, is to make artificial photoreceptor for specific cell types within the retina using optogenetic tools. These are called optogenetic tools. It's a fancy name for a light-sensitive protein. Okay, because I was doing a lot of experiments every day, it was almost trivial to say which out of the 100 cell types of the retina we should target. Okay, so this drawing was coming from 2002, indeed, end of my PhD, when it was you think through what you can do is very clearly there are 4 possibilities you have. And I don't want to go into the science of the retina, that's my goal, but just mention there are 2 opposite ends. So one idea that you can say, what is the absolute simplest you can do? And that is, the optic nerve is formed by so-called ganglion cells. And this is the end of the retina. So you can say, if I make that cells light sensitive, likely they are the ones that die the latest. So that's the simplest to do. And we will talk about later. What is the best to do? And the best is absolutely clear. You go to the remaining photoreceptors that lost sensitivity, but the cell bodies, these cells has a little antenna, which is a light sensitivity in your eye, and then a cell body and an axon to it. And I will talk about it. Turns out that some cases the cell body remains alive when you're completely blind. And then the best one is to resensitize the cones, some of the photoreceptors you have in your eye to light. Because in this eye, the whole retina remains computing, and there are some other reasons that are more complex to it. So let me summarize what was the thinking and asking important and answerable question. What were the properties? So first time is that at the time when I asked this question, I already spent a lot of time thinking about how the retina is working. So there was a deep understanding of the material. And I asked myself, that given that understanding, what is the next step? And that time I concluded the next big step forward is asking this question if we can restore vision. Now, so this was the important question, is that there was simply no therapies for photoreceptor-based blindness. And there is a very important question in science, is this question is answerable? And I'm not saying that you should always worry about that. So mathematicians who worked on proving Fermat's theorem, for example, it was 300 years, it was not proven, and yet people tried and tried. So so I'm not saying, but nevertheless, for an experimental scientist that I am, if you ask questions which are never answerable, you will not get funding from anyone. So it's more a practical way of thinking. It's not necessary, but to me it's kind of important that at one point it's doable. And here the understanding was that, look, there is this gene homology, and people put it into other cells. And any cell you try, it worked. So it must work in the eye. It was a rock bottom simple feeling that one has to work at one point, because it was not too complex. In medicine, if you do too complex things, it just don't work. They don't work, but somehow if it's simple enough, there's a chance it may. Okay, so then comes a second step in this dictionary, after you've done your homework and thinking, is to planning a series of experiments. And I really think that you always have to think about 2 kinds of experiments. One is short-term, another one is long-term, and they have different properties. So the short-term experiment in this case will do a proof of concept with some animal model. A mouse was the simplest one because there were some blind models of the mouse, so it was easy to get. And the long-term, of course, it tests in humans. That's relevant for the goal. But these 2 different kind of experiments, again, 20 years ago, had different properties. So the short-term, I think what's really important, the design, how is the design of an experiment? So we haven't started an experiment, but one important point that all the outcomes are really clearly phrased and analyzed. So one big danger in biology, that it's very easy to do an experiment, and every experiment gives an answer. So very different from physics. In physics, you have to apply for experiment, and it takes a few $1 million to have 2 neutrons to hit each other. But in biology, you just do experiments. And this is a big danger, that people just do experiments. And I like to be careful about that, because I think most experiments can give larger uncertainty after it's done than was before. So what's the goal of an experiment? To decrease or concerted knowledge of humans, decrease uncertainty. But so here, it's very important to analyze that what are the outcomes. In a good experiment, no matter what the outcome, you learn something. So you have to really think, okay, what happens if this is the outcome? What would I learn? What can I tell my mother? It's very important to be able to tell your mother that simplify, and this is an outcome, and therefore this is what I conclude. If the opposite comes out, what did I learn? And every outcome, you must learn something. That's a good experiment. And there is very importantly, that the choose of the right technology. So there are certain technologies can give you a good answer, others not. So thinking about technology. And finally, as an experimental scientist, it's quite good if it's actually doable. So what was the experiment at the time? I concentrated on restoring vision based on targeting cones or bipolar cells in the mouse model. So this was the short-term goal. The longer-term goal is completely different, I think, the way I think about long-term experiments. That I think it's very important to have a clear idea which way you go, but you not necessarily have to worry about if it's doable today or not. 20 years ago, I remember I went to a conference, an ophthalmologist called Arvo, and I presented my idea, it was 2000, no, it was a bit 15 years ago, 2007, presented this idea, and people very kindly, they walked by, and somebody told me, you just lost your mind, that to put some algae into humans. And they were really kind, but also, you are a bit cuckoo, but fine. So it's not necessarily you have to have the technology, because technologies come by. Partly we, in your own laboratories, invent new technology. I particularly like to invent new technologies, but also others do it in the world. So you somehow have a feeling where the world goes and predict, but it's good to have not to know all the details, because you know that in 20 years or 10 years it will be obsolete. And the other one is maybe to talk to others and kind of smell when is the right time. You kind of have an internal feeling now, this is the right time when to go for the big one. And again, I think one point is good to do something as opposed to think about it. On the long run, the long-term experiment was, of course, to do it in humans. And we spent a numerous, with José Sahel, my clinician colleague I worked for a very long time, is in Paris. We thought almost every year for 15 years, when is the right time? And I will talk about that later. Okay, so at one point you think about experiments, but then you come to the third point, which is execution of experiments. And here, the rules are very, very different. So here, really things that don't matter for your thinking start to matter, like good manual skills. So people actually who do experiments have usually good manual skills. So it's good to train yourself. The other one is truly the joy of doing experiments. So something I feel that good experimental scientists, they really love their everyday work. It's that they go to the laboratory, and somehow I, for example, really loved that it was very dark in my room. I don't know why. For me, it was very exciting because, do experimental retina. It must be dark because you work for photons and you want to know how many photons you have in your room. So we have complete dark room, all sorts of goggles on us. It looked like, when I was very young, it looked really amazing. It calmed me down, and I felt myself in another world. And yesterday, I was doing together some experiment in a human retina with Timea Meyer at Semmelweis, and I felt the same kind of really joy that finally there is a retina and we do some experiments. So that is, joy is an important, precision becomes an important point. And the other one, which often underestimated, is work a lot. So somehow the word workaholic get a bad reputation, but I think it's a mistake. I think that being a workaholic is the best ever you can do in your life, because because people talk about the balance of life and work, and I think this is a very misleading thing. And I think the best for a scientist, if your life is your work, somehow it's incredibly satisfying intellectually if you have somewhere that you don't have to worry about all the nonsense around in the newspapers and around, but you have a word that it is real value. There is value in science, very little value of X and Y politicians here and there, whatever country is saying this and that. So I think that there is an immense value and interest in a scientist's life. And I think that the more you work, the more happy you will be. That you are. At least for me, it's 100% true. So another, I think, important fact that in a laboratory, it's always good to work with others. And I don't know why, but in my laboratory, I often find people, when they work two or three together, they're only much better than the ones that work alone. There is something about sharing your daily expertise with someone, with a friend. You somehow choose someone, your pair, and you can discuss your thoughts, your experimental problems. It's nice to work with others. So here, what we have done, it took a while from the idea, but this is what the first proof of concept of cell-type targeted optogenetic visualization in mice. This is a short-term experiment. We set up an experiment model of blindness. I had no clue, by the way. So when I started my laboratory in 2005, I had no clue that what kind of mouse models there are, but a postdoc in my lab had. So it's important that you are not alone. You can get advice if people are working on you. Then we record it from cones. We can show that we can restore activity, record it from ganglion cells. We did behavioral. Again, I've never done behavioral before. But you have people in the lab, and you put together your heads, and you come up with experiment. So at the end, we had a really nice proof of concept for this in 2010. And then comes what I think is the most essential part of scientific discovery. That is the difficulties that you just discover that what you want to do, you run into a wall, and that's the nicest thing in science, I find. So this wall, when you run into it, this is reality, and the true scientist, when you run into a wall, is the one who says, okay, now I did a lot of things, seems to be the word all against us, not going to work, and then you stand up. And you stand up systematically by thinking through what can you do, and sort of build up yourself. And that's what makes, I think, a real scientist a real scientist. So what was the trouble? The trouble were that at the very beginning, when we came up with the idea how to restore vision, we didn't worry much about how to restore vision. And in order to, in a human, you have to inject something to the eye of that person. For this, we need gene therapy vectors. And the gene therapy vector that many, many people around the world worked on, called unano-associated viral vectors. This is a little ball in which there is a little DNA, this is a single-stranded DNA, that coding for the gene, for the sensor, and there is a part we call promoter. I will talk about later. But what was the process? So this is a field that a lot of people, including us, worked on, is developing gene therapy vector to deliver a gene to some cells in the eye. And then we realized here, unexpected difficulties, is that the promoter is a little sequence, DNA sequence, which you can think about as a postal code. So if you would inject in the retina, in the eye, sorry, you inject into the eye a gene therapy vector, if you have a promoter, it can ensure that it expresses the cargo in a specific cell type, 1 out, let's say, of the 100. But turns out that the cone promoter we developed in mice did not drive expression in humans. So that's a bummer. Second one, the capsids, the ball that was optimal for mice were suboptimal in primates and humans. And finally, even worse, the sensor, it came from algae, that worked in mice did not work in primates and humans. So pretty much none of the components, we spent around 8 years or 10 years to work on it. And we basically made this very, very silly assumption that mouse is like human. And fortunately, researchers do these nonsensical conclusions, that they use an animal model, and they kind of imagine that what if mouse would be human? They are not. But we kind of were very happy to cure mice, and we were very good at curing mice. So I remember a postdoc came to my lab and said, oh, this mouse was not blind. And I swear you it was blind. We just treated them with our optogenic sensor. But turned out to be that was useless. And that was a big learning curve, and we lost 10 years there. So the 10 years was that we now systematically had to figure out that how on earth now you make a promoter for human, how you make a capsid for human, and how you find a sensor that works in humans. So we needed to develop a completely new generation of gene therapy vector and test them in a new generation of models, because suddenly we needed human models as opposed to mice, and we needed a way of creating new vectors. So I'm not going to go to detail, but it really took around 10 years, and we published recently this 10 years of work in 2 papers, one in Cell and another in Nature Neuroscience, where we were based on the human retina's single-cell gene expression atlases. We are now able to make libraries of gene therapy vectors that we can directly test in humans. But okay, where is the human retina? And here really I think I have to give a lot of credit to Arnold Szabo, my colleague in Semmelweis University, who kind of mastered the way of keeping human retinas alive. So the first idea really came from Serge Picot in Paris, that okay, we need to test our vectors in human because we seem to have run into trouble. So here is idea to culture human retinas in a dish post-mortem. So these are coming from organ donors. When you remove the eye, the person is dead, you take the retina and hoping that you can keep the retina alive for a long time. So he was able, Serge was able to keep retinas alive for around 3 weeks. It's not enough to test a gene therapy vector. And Arnold Szabo, working in Semmelweis, and I had no clue that he's working on it, mastered this technology that he can keep alive retinas for like 3 months. And this is really, really, it changed the way that we do translation. His technologies are used around the world. And it's something, again, that I think, if you imagine that somebody, I don't know the Hungarian sources of funding, but let's say in Switzerland, it would be the Swiss National Foundation. And we would give a grant that, I want to spend 10 years of keeping the retina alive. Who would give you funding? No one. So there is a problem with science funding, is that the way we judge is always like whatever, where you publish. And sometimes it's good, sometimes it's not. And I think it's good to have very different funding sources and listen to the problem. And sometimes think, it's a very important problem. And maybe the solution will not come fast, but it's worth to support. So Arnold Szabo is such a person, a long-term runner, who worked on this technology and really changed the way the world is doing, can do now research. So we could test actually using his technology, gene therapy vectors. And then very fast we found now different gene therapy vectors with synthetic promoters that can specifically drive different cell types. So here, just images of different retinal cell types. Again, the details are not so important. The second technology, and there is one problem with the post-mortem human retina, that very few of them are available to us, right? And if you do optimizing gene therapy experiments, you want to have thousands of human retina in your hand. And this came, another technology that Magdalena Renner developed in my lab, is from skin cells that was reprogrammed to iPSC cells, and using a technology to push them into the becoming like an eye. Magdalena Renner developed a technology that now we can grow human retinas in a dish to a level that they are actually responding to light. They have 5 layers as a normal human retina, and they have basically all the cell types of the human retina. And we can make now tens of thousands. So we just did an experiment with 20,000 human retinoids, organoids. And so now with the first time, for example, with Novartis, we did the first drug screen on a 3-dimensional organ where a single cell type is labeled, and we deployed thousands of drugs to look at, to find some molecule. So this technology, again, completely changed the way we do things, because we have ad libitum. From any of you, I can grow retinas in a year, ad libitum. And this is important. So one, you have the true human retina, but now in the laboratory, and we have robotics and everything for this, is that we can do experiment mass scale on human retinas. Okay, so these were the challenges get slowly, so we return now again to experiments, but now in humans. And the disease we have been interested, the primary disease is what is called retinitis pigmentosa. This is a genetic disease. It's not one disease, it's a collection of diseases that look similar. And namely what happens here is that these are monogenic diseases where you have a mutation and somehow, and I don't want to go into the details again, just imagine that the photoreceptors lose light sensitivity, and at one point a number of patients becoming blind, but in the whole retina blind. And there's another very, very interesting and much bigger disease called age-related macular degeneration, a phase called geographic atrophy. We are also sort of working now is a next generation of therapy towards geographic atrophy. But these are the diseases. And for a reason that I explained to you first, we choose to go to humans targeting ganglion cells with José Sahel. And you can ask yourself, but why you go for ganglion cells if you explain to me that cones are the best to run for? And this has to do with a lot of timing, history, randomness, and partly, for example, that at one point we felt that now the technology is safe to go to humans. And we had a small company that was ready to go, but we were not yet ready with the right gene therapy vectors from cones and bipolar cells. So partly it was simply we were not ready in one hand, but we were ready to do ganglion cells. And we concluded that the reason to go for ganglion cells is that we can learn more from the patients than we can learn in any experiments we do in the laboratory. So if we do the first experiments and it's safe, the patients will tell us what is the limitation of what they can do. From now, it's an experiment that we can do the next step after. So what is this? How we do it? We simply called intravitreal injection. This is just an injection to the eye. It takes 10 minutes to do. It's really simple. And we had to develop a medical device. It's a goggle that has a video camera that's watching the world. And it is a little projector that projects something to the eye. So that took a while, and there's a little computer to it. And the reason we needed to do is two. One is that humans, we have the immense capability to adapt to light. So if from this room you would go down to the cellar right now, in few minutes after you would be able to see. Now, if you would, from this room, you go to the sunshine, initially you will be blinded, but see again. So the visual world has 10 logarithmic units of distribution of photon intensity. But algae proteins, these algae proteins, are able to see only across 2 logarithmic units. Okay? So you needed to adapt to differentiation. This is done by this goggle. And we can also provide the right wavelength, and there are other interesting properties. Okay, so now finally, after 18 years, we arrived to this patient. And I will tell you 2 patients. We have currently 5, and I will tell you what can you do now if your ganglion cell, that the output cells of the retina is light sensitive, but the retina is not computing because they don't get input. Okay, so, and there are very interesting observation you can do yourself. And first, we test these patients without a goggle, so that means that they don't have the stimulation light. Maybe just to mention one point, that this patient is 58 years old at that time. 2 years ago, he went blind, legally blind at age 48 as a French patient. And he has a bare light sensitivity. That means is able to say where the light is coming from, but unable to find object. And in this is the control experiment. There's no goggle, so he should have it's a normal blind condition because there's not enough light to activate the sensors. Opa. We go back. Up. Okay, we go back here. Maybe we're somewhere here. Yeah. Okay, so here's a patient. His task is to find that notebook. He says he feels the light.
Au bout d'un certain temps, je ressens qu'il y a une différence d'intensité. Mais sur la table, je ne vois pas.
So that's the state they just don't see object. And then we put on the goggle. And now watch what happens. So first of all, you see that the patient starts to scan. You see, by the way, that the camera sees on the laptop. So that's where the camera is looking at. So first what you see is scanning, partly because the retina has almost a small light sensitivity. And at one point, he puts the, now it's on the middle of the camera.
Et alors, c'est où? C'est...
Pardon, c'est sur la droite.
OK.
Okay, so he senses it, he tells you it's on the right, and incredibly precisely puts on it. So what did you see? So a couple of things you have seen. First, it took a long while to find the object. Why is that? The reason is because it's very small part of the retina made light sensitive. I will actually show you which part. So that's understandable. But then what is really interesting is that once he finds the object, which took a while, immediately the motor reaction is incredibly precise. That means that the motor reaction is stored in the brain independently that you had 10 years of blindness with an incredible precision comeback. Now I show you an even more striking example. This lady now counts pencils, or pens, sorry. So she's speaking in French in the background. Again, you can see the camera. So it goes through 1, 2, 3. And at the very end, it says,
Et le bord de la table du fond est où?
it's not very loud. As there is the edge. And now she has to point on it. Look at the motor reaction. Bam.
Parfait.
Okay. Wow. This patient had zero light sensitivity before. So one of the one that what we call is completely blind, yet the motor reaction, the precision. Okay, so the question comes next. You did a lot of experiments, and you arrive to the fifth point, which is you have a lot of data. So when you have a patient like this, you can look at it and you can be happy, but at the end it's data that matters. So I'm not going to talk about how we analyze this patient's data, but generally I'd like to talk to one of my favorite subjects, which is data analysis. And the game is how to turn numbers to knowledge. And this is a wonderful game. So if you think about that any experiment just ends up with number, and your brain has a knowledge, you have a model of the world, and from the numbers, you have to update your internal model. So this is not a trivial thing because you can look at numbers and say the numbers are not talking to me. There is a very deep human, long time understanding how to turn numbers into knowledge. And it has really two components of it. One is logic. And I think that we believe, logic is given to us by nature. But I don't believe so. I think logic is something that's learnable, and logic can be complex. You know, when I say A and B is true, if A and B are true, you can always say, well, of course, if A is true and B is true, A and B is true. No, but what if you have, a lot of ands and ors and not and something, maybe 100 of them? It's not so easy to look at it. So there are rules in logic. And to fully understand the existential quantifiers like exist or uniquely exist or every exist, and then combining with logic, there is more to it. So that's one element is logic. And another one is really the world works probabilistically. So you need a probabilistic understanding that everything can happen with a low probability. And that's a problem because you have to always know, was it by chance or is actual understanding? So over the years, when I kind of see students and try to tell them what to do, I recommend for young people to really look at 4 different things, which you can do much more than that. But these 4 is really essential to turn numbers into knowledge. One is logic, and one of my first book, I learned logic from, and that followed by many others. But this is by Quine, The Methods of Logic. It's an incredibly eloquently written book, understandable by any student. And it's a really great introduction that, logic is a complex thing, and there are ways out. There are ways to understand, actually, human logic, how we apply to very complex situations. The other one is statistics, of course. This is what we use in, and I love to read statistics originally from the people who wrote those original ideas, But again, very early on, I found a book which is very good for high school students, and this is shown here. But many people believe that statistics is the end. Oh, we know statistics and it's all fine. But statistics is not mathematics, it's art. And it's an art because, you know, we say it's significant. What significant means? It means 1 out of 20 cases, it's not true. Okay, so it's good or not good. I say, okay, I'm better than you. It's like, okay, 1 out of 100 cases, it's not true. But, you know, it's our choice. But there is a science behind it, and it's called probability theory. So all of you who are truly interested in understanding of how, you know, has to be thinking in experiment should really start a probability theory and do some statistics, but that's the essence. And probability theory, this is a lovely book that I give it to my students. And finally, what is essential today is programming. And because that's how you turn numbers into ideas, this is where you analyze. I very much advise people against using all sorts of packages. Rather, the best is just program what you want to do. And today, the best I hear from young people is Python. I'm not young, I'm old. I'm using Mathematica, which was written for mathematicians. I don't recommend using that. But it has an advantage of, it has symbolic language as well, so I can combine their logic and data. But by any reasonable way to go, it's Python today, which is it's free for everyone, and that's the way to go. Without programming, I don't think that a young person today can do science. Data will become much more important when I was a graduate student, because the masses of experiments we do, more and more young people, they do an experiment, and the next 2 weeks they analyze their data. Okay, and that's my last point before I finish. And this is something not spelled out quite enough. But once you finish your experiment, you get your data, you wrote your paper, there needs to be silence in your brain. And that needs to be to update the way you think about the world. So we have an internal model in our head. Everyone has an internal model. For example, people talk about hypothesis-driven research or not hypothesis. It's a nonsense. There is no such a human that doesn't have a hypothesis. That's internal to brain to have a model of the world. So, but you have to update it. You get new results, and he has to think through what are every single consequence of these results that comes to you that requires time. So one, for example, is that how you update your model, but then what is the next question? What is internally coming from what you learned as the next question? And in this case, we had 5 patients tested. They had good object recognition, but no face recognition and no reading. So why, the elementary question, why you have no face recognition and no reading ability? And I like to explain it to you something that we underestimated at the time. We knew about it, of course, but we underestimated the importance of a structure in the human eye that animals, so most mammals don't have it, only primates have this structure, and this is called the macula, and the center of it called the fovea. So around 0.2% of the retinal surface, there is a small structure here shown. This arrow shows where the nose is. This is the optic nerve. These are the arteries coming into the eye from the optic nerve. In this area, you can see there's a little discoloration. This is a macula. It sounds like whatever. There is a millimeter area. Turns out to be that area is the most sensitive part of an entire body. So much of your human brains is doing nothing than looking at this at a 1.5 millimeter diameter structure, because there's a massive number of photoreceptors, massive number of ganglion cells that all went this area to create a high-resolution image of the world. And this high-resolution image, what actually you call vision. So if you lose your macula, you think you're blind. And we have here the chairman of ophthalmology who can tell you that people with macular degeneration, they are desperate because they cannot read recognized faces, but most of the retina is completely normal. It's just that this is what we use for high-resolution vision, and more we want high resolution. That's why you have your cell phone where staring. Whatever you do, you want high-resolution vision. If you lose that, you're out of society. But what is the problem in here? So why these patients don't recognize face and read? So this is the fovea is for recognizing faces and reading. And the answer is twofold. So I'd like to explain it a bit more. So one is that there is no computation in the retina. If you make the last cell layer of the retina light sensitive, the retina loses all of its computation. That's one. But a second is even more fundamental, is that in the fovea, the structure of the retina changes. So the photoreceptors, here you see them, are on an array. It's a two-dimensional array like a digital camera. But unlike in the periphery, the ganglion and bipolar cells, during development, they are shuffled to the side. You see, they are shuffled here on the side. Normally, they would form a layer, but in the shuffle, so they are on a ring structure. So if you make a ring structure light sensitive, you will never be able to read it. You can try. You can have a camera which try to kind of distort in 3 dimensions. Then you will get a crooked image. And this is shown here. Is that what happens that if you, at the very center of your eye, you make ganglion cells light sensitive, you have a light sensitive torus, but then nothing at the center, so you will not see what's the face in the center, and the whole image gets distorted. And if you make the cones light sensitive, these are on a camera, they are on a mosaic, and therefore you can get actually a high resolution vision. So this is how my institute started, that we recognize slowly that what we really have to do is go back to the original idea and make cones light sensitive in the fovea. And this is a collaboration with Hendrik Scholl and Bence György, who is an incredibly talented Hungarian researcher. So we had to, again, design new viruses, and in 4 years, find one which really exclusively, with exclusive precision and almost 100% efficacy, targets human cones in the fovea. And this is sort of like the last few thoughts. Then how do we know at the end, how an experimenter know that we can restore normal human retinal activity in a postmortem retina? So here is a technology, is that on the very first day, when on postmortem, on the very first day, we can actually record normal human activity from a donated retina. So we have a little chip here. It's made in Basel by a friend of mine. It has 30,000 electrodes. You put on the retina, and we project images on it. And it records from the ganglions as the message that goes to the brain. So here, for example, we are kind of wiggling light, and you can see that 1,000 human ganglions are responding to it. So precisely on the very first day, we can read out the message that the optic nerve sends to the brain. And we have 12 hours to do that. And after, the retina loses its normal light sensitivity. And then we come with the gene therapy virus, and we restore light sensitivity, and 5 weeks later, we again record with the same multi-electrode array and compare side by side how is normal vision that goes to the brain versus the one that we recovered. And again, I don't want you to understand it fully, but in the center of the human retina, there are 5 types of responses, and precisely the same 5 types comes back after optogenic therapy. So we know that these patients we will be treating will have identical message coming from the retina to the brain as in normal. It doesn't mean that they will have the same vision. There's a lot. It's not going to be the same vision. But this is, I think, the best today one can do. So I'd like to kind of finish here, and this is what we try to bring to as the next therapy. So I'd like to finish with this slide, is that I talked about the elements of scientific discovery. I think there are 6 major elements. One, where everything starts to do with thinking, asking an important yet answerable question. The second is about planning experiments in the short and long term, which has different rules. Executing the experiments. Importantly, all of you in experimental science, we run into difficulties, and you have to find ways out. I talked about data analysis, which is becoming more and more important as experiments will result more and more data. And I think the real winners of the next generation will be those who truly understand how to turn data into understanding. And finally, it's very important to update the way we think about the world and asking follow-up questions. So with this, I'm very happy to answer questions, and thank you very much for your attention.
Thank you very much, Botond, this brilliant presentation. It was a privilege to listen that. So now the stage is for the students. The stage is for the students. Now you can ask questions, so question time.
Oh, thank you. So you said that your researchers did rely on others' ongoing researches. Can we say that these kind of relations and knowing about others' researches are doing the base of your research? So it is like a gambling that is there that kind of researches that you can rely on?
Well, look, so it depends on your age. When I was a graduate student, I was working alone. Actually, I was a single person in that laboratory after a while. But as I started my lab became bigger, and today there are maybe around 30 people in my lab with whom I work together. So in one part, a laboratory has many people in it. It's working together. So that's one thing, what is in your laboratory. But what I meant on the long-term experiment to sort of believe that people will do research for you, I think it's very important to watch your field and be in contact with your competitors. Competitors in science, I think, is very friendly. So it really, I think competition in science is a little bit like sport. Is that people go, they run, one wins, and it's okay, and it doesn't really matter. I think competition is generally gives adrenaline, it's good. In science, it's a friendly competition that, no get punished by not winning. But nevertheless, this is ongoing, and it's very important to watch the world. Now, when you're young, the way you watch the world is via publications and critically assessing. The problem with publications is that much of it is not true or irrelevant or other problems, and others are really, really very good and very important. So you have to kind of develop with your supervisor and your own self what you judge. And I think the best way to judge is looking at your own data. Is that your own data, you work on something, the data tells you what is true and what is not true, but start to read other people, not to follow them, I think is a very, very big mistake to follow others. And I can illustrate you by imagining that you want to win the 100-meter running in an Olympic game. And somebody tells you the best strategy, always follow the guy ahead of you and try to keep the pace. You can see that's not going to work, right? You can get second or third, but not first. And that's a big difference. So the big difference of people who kind of try to see what others do and they do it, or you sit down and you ask, what is the most important question you ask, and follow your own, what your heart is telling you to do. Nevertheless, you always have to be aware that there are others as well, and it's a very competitive environment. And you might be scooped anytime, and it's fine. You go on, but I think you have to follow your own judgment. But that's what I mean by following others. Also, my experience that when there is a new technology coming that has use, the world jumps on it. And when, when there are hundreds of researchers jumps on it, they will develop something quite. So you can rely that the community is much stronger than an individual or an individual lab.
Thank you.
Other questions? Yeah.
So my question is, will it ever be possible to determine the DNA or the gene of the human photoreceptor protein and insert that into a cell in case there is no post-transcriptional error?
Yeah, it's an excellent question. The reason it is difficult, because in a human cell, I told you that humans, we have this immense capability to adapt to different light levels. And that requires not one gene, but at least 20. Inserting 20 genes today is completely impossible. So the human is a G protein coupled receptor, and you put in a cell, you don't know the state of the disease cell, what it couples to. So some people try to do it. I don't believe that's the right way to go. I think, but maybe one day it will be possible. There are other ways also to think about the problem. The best way would be not to lose vision, right? We are very bad on that front, unfortunately. It's actually much easier. We have technologies to restore vision, but we don't have technologies to slow down vision. In some, very small, so for example, in macular degeneration, there is a so-called anti-VEGF antibody that can help, but these are also just delaying the problem. But completely stopping degeneration would be the best way to go, and that is not, we don't know how to do it today. Unfortunately, inserting a human genes, you would need a lot to work on it. Yeah.
Next questions.
Thank you. I would like to ask a rather personal question. How did you decide on going with medicine rather than mathematics?
I didn't understand. How did I decide to?
To rather finish your studies with medicine than with mathematics.
Yes, it's a very good question. It had to do with my father, who was a very strict man. And he always said, what you start, you finish. So I offered him to finish medical school quite earlier, every year, for example. I offered that, why don't I finish medical school now? And he told me that, well, you started it, you finish. And also, he had, yeah, that was it. So, by the way, I wanted to leave science also many times because, when you run into a wall, you think, you go down. But at the end, somehow, my curiosity always prevailed about the unknown. And I think that this is the best I feel, is that when there is unknown, it raises adrenaline in us, that there is a chance to get solved. And I think it is an incredible pleasure inside, which is, I think that if you work enough at one point, is really way higher than getting good food or going to whatever island to vacation. Yeah. Maybe one thing I forget, and that's just it's this. And just any of you interested to do PhD, we have a PhD program that is closing. So we started a PhD program. We get a huge donation from an individual. We're very thankful. And we're starting a PhD program, which I think it closes November 18th, it's closed. But I don't know what stage you are in your career, but any of you, we have few positions, not many, but few positions fully funded. So this is just an advertisement we are really open from mathematicians to physicists to anyone who is interested to think with us. Thank you very much.
Thank you. Any more questions? First there, and the mic comes here as well.
So my question is that in these kinds of diseases, when you lose your vision, what is the reason that the loss of vision occurs in the first place, and if that reason won't make your therapy obsolete in the first place?
Yeah.
Or something.
Yeah, so I'm not sure I fully understood the second part. The first part, I can do it. So there are 70 different genes that with mutation you can get retinitis pigmentosa. And you have two kinds of photoreceptors. One are called rod photoreceptors and cone photoreceptors. The rod photoreceptors are for very low-level light vision, and the cones are for daylight vision. You know that your cones are working if you see color. If you don't see colors, that your rods are working. It turns out that humans really don't use much the rods, only rods, because we always turn on the light enough that we want to see colors. So what happens in this disease is that many of the cases, not all, but many of them, These are genes that express in rod photoreceptors. And because you don't use your rods a lot, you can lose a lot of your rods and you don't feel that you are losing your photoreceptors. Your cone vision is normal. But at one point, a mechanism that no one fully understands, the cones, or they don't express the disease gene, they start to lose their light-sensitive antennae. And that's when patients come to be seen and they are losing their vision. So the reason actually we go for blindness and not for gene replacement, for example. One is that gene replacement, except of the retinal pigment epithelium, very rarely works. In some cases it does. But many cases, the patients who come to the clinic already lost their rod photoreceptors, so there is no genes to replace because they are expressed in rods. The second one, I didn't fully understand your question about why the therapy would be obsolete.
Well, I was thinking that the same problem that caused the loss of vision in the first place would sort of interfere with your receptors that you implanted in the eye. Or
you mean if there is remaining vision that would interfere, or that's what you say?
Well, so if you use your therapy on somebody and gave their eye light sensitivity again, if the problem could make them lose that.
It's fine. So I tell you that two problems can happen. So one is there are two problems can happen. One is that there is a worry that these patients have some of them has actually extra light sensitivity for unknown reason. And it was a worry that you provide a goggle with high intensity, that will become a problem. It's not. So this we just learned it's not. The second one, first, and so this is red light we are showing, or amber light. The second one is that there is a problem with interfering normal light sensitivity, and that is in macular degeneration. The reason we are going for fully blind people, because they have no residual vision. That's the easy case for us. Now, the hard case, if you have a macular degeneration and you lose the center of your vision, and the periphery is light sensitive, then we start to, now you have two vision and we produce, that's more complex. So that's why it requires much more time to think through, and we are not fully there, but that requires much more time to think how you do it. There are ways out, but that's a more complex problem.
Thank you.
So I have two questions actually, and the first one is about how many injections does a human patient require, and how long do they keep the ability to have the vision? And the second one is in the videos that you have shown us, after the patient has recognized the item where it is, they have moved their right hand next to the goggles, and after they were able to reach for the item.
Very good eyes you have, and so it's very important. So the first question, it's one injection, and interestingly AAV, although it remains episomal, so the DNA will not go into the genome, it stays next to it, but in animals, when we and others test this for 10 to 15 years, they stay there. So it's a one-time injection. And there are ongoing experiments now in gene therapy already in clinical use. As a drug called Luxturna, which is injected to the eye. So probably in 10 years we can have experience, but let's say in monkeys and dogs, it's around, people could not see any decrease of the therapeutic value. Now the second question, why the patient did that, as I told you that in the retina, there's only a very small part is light sensitive. And these goggles, as you could see, is not the final design. They were kind of manufactured in small scale. And they are not very good in a sense that, for example, in this particular case, it hurt the patient's nose and it keep adjusting goggles. But there is one more to it. Namely, at one point, he saw that there was an object there. And he wanted to make sure it's stable. The sensation is stable. That's why he adjusted. Okay.
Thank you.
So it's a very small area. Imagine that you have a little tunnel to the world, and you go there, and now as you see something and you want to make absolutely sure that it's a stable image on your retina.
Thank you. And the very last question.
I want to ask you, how could a scientist have a family beside this workaholic lifestyle? Or is it possible?
So yes. So answer it with my father was workaholic, truly. He always denied it. He told everyone his Sundays, he was religious. So that Sunday he told people that it doesn't work. That's absolutely not true. It was a pure lie. So, but what, for me, even until today, he was the person who taught me most in the world, and the biggest influence ever a human was on my, I don't listen to just about anyone, but I really, really had a close relationship with him. And our relationship was mostly scientific, and he worked all the time. But the little amount of time we spent, I learned much more, I think, than many people can learn from their own parents. So the dilemma is the following, that a parent can decide to spend a lot of time with their child, but there's a lot of time what they would do if the parent doesn't have an intellectual insight, right? So you can say what it will do is, let's say, eating together and doing this and that together. On the other hand, if you're a researcher, you get a lot to say, and in this little time you have, it's a miracle for your child. So I felt this miracle from my father that he worked in Berkeley 6 months a year, and I was here as a student. But the time I spent together was so incredibly important for me. So I think that we overestimate the amount of time. And I think that's much more important is that in that time we spent as a young child with a parent, what do we learn? And that's my answer to that. I think that there's a lot of things compatible, except you have to drive your life that way. You can also learn from your children. So it just that I think scientific life involves a whole brain.
Thank you.
Also, maybe one more example. I had a teacher at Harvard. Her name is Connie Sepko. And she went home every day at 5 o'clock because she had two kids that were just born. And I asked her the same question, how come you keep track? And he says, look, I'm with my child, but at the back of my brain, I think all the time. So it's not what you do exactly. You can go running, or you can do the question how much your mental energy is spent thinking about the problem. And it may be for women who go home at 5 o'clock and they are children, they have an advantage because they think more than the men who are in the laboratory and doing experiments all the time and don't think. So I think that the point is that it's invisible sometimes. You can do a lot of things and think at the back of a mind, an incredible mental activity, try to think about the outcome or predict your experiments.
So thank you very much for the lively questions. I would like to hand over the microphone to András Varró.
So finally, we thank to you to spend your valuable time with us, give a very fascinating lecture, and we would like to thank you with a medal and a diploma for your visit in Szeged this time.